Hi guys, I just wanted to kind of go over those two lab exercises, 17 and 19, that you did not come to because you're in quarantine. So 17 is on motility determination, and you should definitely read over that material presented that talks about the flagella, the function of the flagella, those terms in boldface, the fact again that not all bacteria have flagella, but that all of them have both S and M rings with respect to gram-positive and gram-negative cell types. But the gram-negative cells have, in addition to the S and M, the L and P rings. You can reference page 121, figure 17.1. They talk a little bit about that. We did not do the wet mounts or hanging drop slides sections on 122 and 123. But we did inoculate two different types of media, and I want to show you those. These are not mentioned in the lab book. The first one is a plate called an eye plate. This is basically a Petri plate that is divided into halves by a little partition, and Deb made up some motility media that the students inoculated two different bacteria into, and these are the two listed in the lab book. Proteus vulgaris is one, and Micrococcus luteus is the other. And so what she did, Deb did, is she inoculated just a small section of each of those eye plates, and she circled those areas that she inoculated with a black marker. And then she came back a day or two later, and you can see what's happened. I'll hold this up to the camera. I hope you can read that. The top is Proteus vulgaris, and the bottom is Micrococcus luteus. And you can see, of course, that the Proteus vulgaris has moved out from that little center circle where she had initially inoculated, while the Micrococcus luteus there at the bottom has not moved appreciably out of the initial inoculation line. So obviously, Proteus vulgaris has motility thanks to the presence of flagella, while Micrococcus luteus is non-motile, does not have flagella. So the students basically recreated what Deb had done using this special motility media. And we'll probably be using this again once we get your unknown and start doing some tests on it. The second material that we used for exercise 17 was another motility medium that we buy. Deb does not make this. It's called motility test media. I don't know if you can quite read that, but I'll just read it for you. It says motility test medium with TTC indicator. Now, TTC is referred to as triphenyl tetrazolium chloride, and there's a little blurb here that Deb put on the table where these tubes were being placed, and this will be back there for you to reference if you ever need to look at this again. But in essence, it just simply says that this TTC that's been added to this tube adds visual enhancement to motility media. It is a colorless dye that, when reduced by bacterial cells, forms formazan, an insoluble red pigment. So what the students did was they inoculated one tube with Micrococcus luteus and another tube with Proteus vulgaris. Now, you know from the previous experiment that Proteus vulgaris is motile and Micrococcus luteus is not. So how do we see that in that particular medium, which is actually stabbed, just like is described there at the top of page 124? Well, here's what the students are going to end up finding when they look at these in another day or two. Here is the test result with the Micrococcus luteus. Deb's got that labeled, Micrococcus luteus. And you can see that the red dye, which is that TTC, that forms once the bacteria begin to metabolize some of the chemicals in the media, that the bacteria are restricted to the stab line. Okay, and I'll just twist this, and you can see it has not moved out from the stab line. Contrast that with the Proteus vulgaris, which is certainly growth at the stab line, but you can see how the other part of the tube, it is pink all throughout, meaning that there are bacteria that moved from the stab line out into the entire motility media. So this is motile, this is non-motile. So that basically was exercise 17. So you really didn't miss a whole heck of a lot. And as I said a moment ago, we'll probably be using one or both of these later when we test your unknown for motility. I did want to talk a little bit about exercise 17, or 19 rather, which is enumeration of bacteria. And I'm going to pause this for a second. Okay, I'm back. Enumeration of bacteria, the standard plate count. What I want to do is kind of walk you through the procedures that we followed this week in lab to do what's referred to as the standard plate count. This is described there on page 139 of your book. So I want you to very carefully examine this, and I'm going to try to share with you one of the diagrams, which I don't see here. Where did it go? Okay, I'm going to stop here and pause. I'll be back in a second. Okay, let me share the screen here and reference that diagram, which I thought was going to be blown up. But anyway, yeah, I think we'll be able to still talk about this, and you can still reference that figure 19.1 of your book. So each student was given a culture, a broth culture of E. coli. And what they did was they transferred a milliliter of this E. coli broth into a water blank, it's called. This contained 99 mils of sterile water. Okay, 99 mils of sterile water in this bottle labeled A. That created a 1 in 100 dilution. So 1 mil added to 99 is 100. 1/100th of it is E. coli broth. Okay, so that's what the 1 in 100 refers to, the dilution that's created. That bottle was then mixed. And let me see if I can again share the procedure here that we used. Well, actually, I'm just going to have you reference. I'm going to go back to this diagram again. I'm going to have you take a look on page 142, figure 19.3. You can read about the actual procedure of how we mix that. So check it out. It's described there on step number 3, page 141. So they mixed the water and the E. coli in bottle A using the prescribed procedure. And then they transferred, using another pipette, a different pipette tip, a milliliter of this into a 99 mil sterile water blank. In other words, they did another 1 in 100 dilution. However, you see this says 1 in 10,000 because we did 2 1 in 100s. That equals a 1 in 10,000. So this dilution factor is representing the original culture. And then finally, after bottle B was mixed using the proper protocol, we transferred a milliliter of it into a third 99 mil sterile water blank, creating a 1 in 1 million dilution. In other words, another 1 in 100. So we've done 3 1 in 100 dilutions, creating 3 different dilutions, a 1 in 100 in A, a 1 in 10,000 in B, and a 1 in a million in bottle C. Then what the students did was they made 4 transfers into agar plates. 2 came from B and 2 came from bottle C. When they delivered a milliliter from B into a sterile plate, The dilution factor did not change. So it came from the 1 in 10,000, 1 milliliter did, and therefore that plate, that amount of E. coli suspension, that dilution, is a 1 in 10,000 dilution. But I want you to notice this additional transfer into a different plate. This was 1/10 of a mil. That in and of itself is a dilution. So rather than a 1 in 10,000, like we had here, this becomes a 1 in 100,000. So we do, again, an additional 1 in 10 dilution from the 1 in 10,000. Hope you understand the math. And then the final set of transfers coming from bottle C, 1 mil transfer into a sterile plate. Again, dilution factor stays the same, 1 in a million, and 1/10 of a mil transfer into a fourth plate, which makes this a 1 in 10 million dilution. These 4 plates are then incubated. They're inverted, placed in the incubator, and the students are going to be looking at those maybe today for the Tuesday group or tomorrow for the Wednesday group. Or if they can't get in the next couple of days, Deb's going to pull those plates, and as well as these tubes and the eye plates as well. All this stuff is going to go in the small refrigerator on the right-hand side of that pair in the back of lab, so students can access this. So if you have any interest in looking at these on Thursday, next Thursday, if you come in to do the practice staining, maybe somebody who's been in lab can show you what they discovered. So when those students go to pull those 4 plates, these 4 plates, they're going to be choosing the plate that has between 30 and 300 colony forming units. That's described again on page 139 under standard plate count. So make sure you read over that. Colony forming units are what we're looking for, and the plate that we're going to choose has between 30 and 300, because those are the statistically valid numbers we have to work with. Basically, then what is performed is a very simple math calculation, and that's described on page 142 on the right-hand column toward the bottom. Step number 3, calculate the number of bacteria per mil of undiluted culture. So we're looking at how many cells per mil are in this original culture that we began with. That's the idea. And so if we had, again, say 220 CFUs, and we had the 1 in a million plate, let's say this was the plate that had the 220 colonies, then we simply multiply 220 times a million, and we express that number in scientific notation, which would be 2.2 times 10 to the 8th power. And I hope you remember scientific notation and how that's done. If you don't, go back and look at your algebra. And we use only 2 significant figures. They talked a little bit about if there was 227, we would round that up to 2.3 times 10 to the 8th. We wouldn't write 2.27, we'd write 2.3. So that's in essence a big part of what we did in lab yesterday and on Tuesday. We also ended up working on the spectrophotometer, which is described there on page 143 and 144 and 145. I am not going to go over that. It's just impossible for me to describe it. You really have to see it in action. You can talk to some of the students who were there who actually did it. Hopefully they could maybe kind of expand upon that, but I'm not going to do a lot of testing on that part of chapter or exercise 19, so don't worry about that. I think you should kind of try to read it over, glean as much as you can from that. But I wanted to just wrap up by talking a little bit about, again, once you come back from quarantine next week, I think if memory serves, you can come back on Wednesday, which by the way is a Monday class schedule. I will not be here until Thursday the 15th. And so I think I mentioned, and if I didn't, I apologize, but I'm going to tell you now that Thursday the 15th, when I come back, we've arranged to have the micro lab open for you, and Deb is going to be having prepared some cultures for you to practice your stains on. because the practical the following week, either on Tuesday the 20th or Wednesday the 21st, that practical is going to be a two-part practical. And I've talked about this to the students who were here this week, so I'm telling you this for the first time. There's going to be a 10-point written component that will basically be a quiz over exercises 15, 16, 17, and 19. So you're all going to come in, you're going to take that 12-point quiz at the very beginning, and then when you're done, you're going to turn that in, and you're going to choose a stain. I'm going to have a container with all the stains written on a piece of paper. So there'll be a simple stain, endospore, gram stain, acid fast, all the 6 stains I think we did. You're going to reach into that hat, pull out a piece of paper, and that will be the stain you will be asked to do for the second hands-on part of the practical. The first thing that you will do is you will write down all of the steps, including aseptic technique, that you're going to follow in making the smear and the subsequent stain. All of the stains are going to be in those small containers, so if you forget the names, you can look at the stain names, that's no problem. But you need to write down all of the steps in numerical order, again, making sure you include aseptic technique. You don't need to include wiping down the tabletop and washing your hands and all that stuff, but let's assume that's all been done. And you might be given a broth, and you might be given a slant, or maybe even a plate. So be able to talk about how to manipulate a semi-solid medium like an agar or a liquid broth. Once you've written down the steps of the stain, you will come back up to me. I will give you a sample, a bacterial sample, either a slant or a broth or a plate, and you will actually then perform the stain that you chose. I will probably occasionally look to make sure you're following a good aseptic technique, but I will not be hovering over you, but I will be watching. There'll be a few questions that you'll have to answer once you've performed the stain and examined it under the oil immersion objective. And I would like you then to call me over so I can take a look at the stain and evaluate the stain, because there'll be some points associated with the quality of the stain. There'll be some points associated with the answer to your questions as you evaluate what do you see when you look at this slide. What's the morphology of the cells? Is it showing a gram-positive or a gram-negative test result? Do you find this to be an endospore former or not? This is all based upon the results of your stain. And then once you're done answering those questions, and I've had a chance to look over your slide, you'll turn that into me. And the last thing you'll do before you leave is you will choose a slant that I will have up in front. It'll have a letter on it. I think Deb's going to label them A through M, or however many people there are in the lab. And I'll be asking you to do 2 subcultures onto 2 more slants. The original unknown I will get back from you, and the 2 that you have subcultured will go into the incubator for 24 to 48 hours, and then those will need to be pulled and placed into the refrigerator so that we can then use those the following week as we begin our battery of tests on the unknown. So I think that pretty much wraps up what I wanted to tell you, both in terms of exercises 17 and 19, and also to bring you up to speed on the practical. The last thing I'm going to mention is the fact that I will have some sign-up sheets. Now this happens to be for the Wednesday group, and I'm going to ask students to come in at various times to begin the practical. So you're not all going to come in at 2:40. I'm going to have you staggered. And I can have as many as 2 people come in at one time. So you can see for the Wednesday group, we've got Mackenzie at 2:45, Lauren at 2:45, Nicole at 3, and Mackenzie at 3. So I'm not sure how many more people are left in the Wednesday group. Let me just see if I can find here real quick who's in the Wednesday group. So it looks like Mike and Courtney and Leah and Skylar and Michaela and Amanda and Courtney. So it seems to be, I don't know, 6 or 7 of you. So I will be probably asking you to come at a certain time. I'll email you, or I'll send out another little video. But we can, figure that out coming up in the next couple days. For the Tuesday folks, same idea. I have started a sign-up sheet for the Tuesday people, and Aiko and Ashley have chosen the 3 o'clock time slot. So we've got a couple 2:45s, we've got a couple 3:15s, and a couple 3:30s. So if you're in the Tuesday group, if you would prefer a 2:45 or a 3:15 or a 3:30, you can let me know, and I'll jot you down in there at those times. So we can talk more about that coming up, maybe next week, or you can stop in the Zoom room and ask me questions. But I think I'll stop there and get ready for A&P Lab that I have in a few minutes. Okay, hope you guys are doing well, and we'll talk to you soon. Bye.